It's my great pleasure to introduce Yuchao Jiang, who is currently an assistant professor at UNC Chapel Hill. He holds joint appointments in the Department of Biostatistics and Department of Genetics. He got his master in statistics in 2014 from University of Pennsylvania, and then he got a PhD in genomics and computational biology from the same university in 2017. He is a PI of the Statistical Genomics Lab in UNC and broadly focuses on statistical methods for genetics and genomics. And I also would like to note that he recently got a Mira Award from NIH and IGNS. And today he would be talking about methods for single cell. Let's welcome our speaker.
Yeah, thank you so much, Alex and Irina, for the very nice introduction. I also would like to especially thank Brani, Mark, and Alex, and everybody involved for making this possible. It's my great pleasure to be here. And I've definitely very much enjoyed my visit so far. So today I'm going to talk to you about our past and ongoing efforts on developing statistical and computational methods for single-cell integrative genomics. Now, I've tried to tailor this talk to sort of both the statisticians and the biologists, but in the end, I decided I'll just go slow, and I don't absolutely have to go through everything. And then feel free to stop me whenever you have any questions, okay? So here is just one intro slide. Heterogeneity really has been the theme of research in biology for pretty much as long as we know it. Now, the recent advances of next generation sequencing data really allow us to study heterogeneity at multiple different levels in a high throughput manner. So here are just a few examples. If we look across different individuals, we see that in this case, different ethnicity cohorts have different copy number profiles. And then if we zoom into one person, and in this case, we look at the gene expression patterns across different tissue in this person, we see that there is a lot of differential expression patterns across the tissues. This has been very extensively studied by, for example, the GTAC consortium. Now, if we further increase our resolution, and we look at the cells that come from the same tissue, we find that most of the times the cells belong to different cell types. And then even for the cells that come from the same cell type, they might be at different cell stage or at different time point in cell cycle. So it is very important for us to disentangle and differentiate these different levels of heterogeneity because it is very much related to health and diseases. And here is just one specific example on cancer heterogeneity. So when cancer genomics first started around 2 decades ago, we were sequencing one tumor dissection per cancer patient. And our priority goal in that case is just to find clusters of patients or types of the cancer. And then very soon we realized that there is a lot of intra-patient variation, even for the patients that have the same type of cancer. So for this quote unquote, personalized treatment, when we look within one individual cancer patient, we find that there is also a lot of intra-patient variation. And now importantly, this variation does not only exhibit between two different tumor sites, for example, between the primary tumor and the metastases, but more importantly, we find that there is also a lot of heterogeneity within one tumor dissection. And then basically, these are what we call the clones, defined as cancer subpopulations that are genotypically and phenotypically distinct. Okay, so it's really those clones that are resistant to treatment that will lead to, in this case, the relapse and the metastases. Okay, so the single cell technologies really allow us to study the variation within one specific patient and then within one specific tumor at the finest possible resolution. So in this case, we'll be able to characterize the molecular level, characteristics of the tumor. Okay. So the bulk tissue sequencing, we pull millions of cells together, and then literally we just read out a mean or an average from this readout. The single cell sequencing, on the other hand, it really have the cellular level readout and it circumvents the averaging artifacts. But on the other hand, while this really, offers really unprecedented opportunities, rigorous statistical methods and scalable computational algorithms are lacking for, the integrative multiomic analysis. So today my talk is going to be focused on how to characterize genomic heterogeneity from two different perspectives using a multimodal and integrative approach. So for the first part of the talk, I'm going to focus on recent framework that we developed to interrogate cell type and cell state specific regulatory mechanism. Okay, so here is just a very brief background. The DNA shown here is composed of some closed region and some open region. Okay, so in the closed region, the DNA wraps around what we call the nucleosome, and then it's very dense. It doesn't really have any interactions with other molecules, proteins, RNA, DNA. In the open chromatin regions, there is this, especially in the non-coding open chromatin regions, there is this specific region that we call the enhancers. Okay, so the enhancers contains bits of the DNA sequences, 10, 15 base pair long, that a protein called a transcription factor can recognize and bind to. Okay, importantly, the binding leads to, or as a matter of fact, is a part of a process in the 3D genome space that loops the enhancer to the promoter region of the target gene that this whole transcriptional machinery regulates. Okay, so in this case, we have the promoter region, which is upstream of the gene, and then we have the target gene here. So this may seem very complicated, but in short, the TF binds to an enhancer region, which is really far from the gene, and it loops to the gene, it regulates. Okay, so it has an effect on the gene expression levels. So in this case, you are going to talk about two different sequencing technologies today. One is RNA-seq, which is really simple, so it measures the gene expression profile. The other one is called the ATAC-seq. So this basically measures how open or closed the DNA is. So RNA, the features is gene. The ATAC, the feature is a peak. So the peak openness, the peak accessibility, or the peak intensity tells us the the openness of the DNA. Okay. So what's really important in this case is that we now know very well that the cataloging as well as the regulatory activities of this transcriptional process is highly tissue and cell type specific. So really, we need single cell approaches for us to disentangle the cell type and cell state level mechanism. Okay, so why single cells? This is just one last sort of intro slide. On the left-hand side, we have the human hematopoietic differentiation trajectory. We see that we start from the stem cell and then it differentiates into T cells, B cells, natural killer cells, and so forth. Now, just around 5 or 6 years ago on the right-hand side, this Nature Genetics paper, so this team carried out very extensive cell sorting. So basically, they find some markers, then they are able to sort the cells into different cell states shown on the left. And then presumably, they just aggregate all these cells that are homogeneous, and then they carry out a traditional bulk RNA or bulk ATAC sequencing. And then what's really important in this case is that they find some clustering information from both domains. And then more importantly, there seems to be some crosstalk between these two different modalities. Now, obviously, single cell sequencing, you don't really need to do this very extensive, very expensive, and very laborious, sorting step. You just, send it to, the library prep machine and then send it to sequencing. You get a single cell level readout. And then more importantly, this would allow us to investigate the regulatory mechanism as well as the crosstalk between these two different modalities. Okay, so these are the technologies that are available. And then the technologies that I'm going to talk about is specifically to generate ATAC and RNA in the same exact cell. You see that the field started around 2018, and then during the past 4 or 5 years, there are these methods or these technologies that are developed. But really, we feel that these are really sort of hand-holded by the labs that can really do this well. Until very recently, 10x Genomics, released a commercialized pipeline where you can basically generate a library using their Chromium, preprocessor. And then you send it for sequencing. So we see that, okay, this is probably going to become really popular. And as a matter of fact, by querying the public repositories as well as through our local collaborations, we do find that this technology is really on the rise. So these are the data that we have processed, including some public ones, PBMCs, multi-embryonic brains, as well as some local collaborations, for example, the HIV latency reversals. So I'm going to focus on these technologies as well as these data sets today. So this is a data format and notation. So remember at the very beginning, I said, it's very complicated, but sort of if you modularize this, there are 3 different factors. Transcription factor J binds to a region, we call it peak region T, and then this regulates the gene expression for gene G, okay? And then for the single cell multimodal input, in this case, your peak accessibility will be read out from the ATAC domain. Your gene G expression will be read out naturally from the gene expression matrix. Your TFJ, of course, TF is a protein itself, but through queries of the protein databases, we find that the gene expression of the TF coding genes can serve as a good surrogate for the TF protein expressions in most cases. So in this case, we use the TF expression, because we have the ATAC and RNA only from this technology, sort of as a surrogate of how the TF is really being produced in the cells. So again, here we have an X matrix and a Y matrix, and then the cells in this case are matched across these two different modalities. For the ATAC matrix, the features are the peaks. For the RNA matrix, the features are the genes, and the cells are nicely matched. And then what we are really interested in is that we focus on specific target gene, your favorite gene in your lab, for example. And then you look in the vicinity region of the genes, usually it's 200 base pairs, kilo base pairs, up and downstream of the transcription start sites of the gene. And then these are the potential cis-regulatory regions that we want to scan for the regulatory elements, okay? And then looking at the existing annotations for the transcription factor bindings, you are able to find the TFs that have a binding motif in these peak regions. Okay, so in the end, what we really want to go over is to find the regulatory mechanisms through this whole bunch of different modalities as well as a different input. So we started with a very simple task that is also carried out in a very standard way by a lot of methods. So that is, can we come up with a model that can make a good prediction of the gene expression from the peak accessibility? So for example, the SIGNAC, SURAD and CSUR methods, basically they just sum up all the accessibilities in the vicinity regions of the gene as sort of what they call a pseudo gene activity. Okay, so this is how they make a prediction of the gene from the ATAC space. And then later on, there's another method they propose, what they call a regulatory potential model. So instead of doing a very naive sum in this case, they were saying, okay, maybe we can use some priors, based on existing genetic annotations in biology. So we do a weighted sum instead. So the weights in this case are prefixed, and then they are pre-calculated from some existing gene annotations. Now, neither method is actually specifically developed for this design. And then there is some methods that try to come up with a sort of supervised framework using the information that the cells are actually matched here. So basically, they're trying to train a model with some penalty terms on the beta coefficient, and then they want to make a prediction of the model that is trained. Okay, but the feature input are the peak accessibility in this case. And then what we propose is that instead of only using the peak accessibility as your features, we use a product of the peak as well as the TFs that we know have a binding motif in the peak. So in this case, sort of, you choose a different set of feature input in this case, and you also do a sort of a lasso regression framework. And then so it turns out that this actually significantly increased our prediction accuracy. And then this is one data set of the mouse skin, and then I show you two different genes. It's quite complicated, but essentially the top row is one gene, the bottom row is another gene, and then each column is one of the four methods that I showed you in the previous slide. So at least for these two genes, we see that if you use the peak as well as the TF, you use the interaction between these two different molecules or factors, it can significantly increase your prediction accuracy. Now, of course, this might seem a little cherry picking. So if you do a genome-wide sort of prediction accuracy assessment, we find that across these three different data sets, by incorporating the TF information, significantly increase the prediction accuracy. And of course, you account for overfitting, et cetera, in this case, okay? But really prediction, can get you so far in terms of understanding the mechanism, okay? There are a few reasons. The major reason from a statistical point of view is that how to make inference and how to test for the significance is non-trivial. Okay, so there's a huge literature in statistics, selective inference. So basically, you need to make inference while account for the fact that the model is not fixed. And also it's actually estimated from the data. So how do you do inference in this case is non-trivial. We feel that the existing methods either are computationally intensive or they lack of power. Now, biologically, we feel that there's an even stronger reason why we don't want to resort to this, penalized regression framework, in the sense that we have a lot of collinearity in the feature space, and then we know that the lasso actually will just randomly pick one feature among the multiple correlated features, But in biology, you want to return a comprehensive call set, especially if you want to, for example, generate a network for your downstream analysis. So we really want to do sort of a genome-wide scan and want to be comprehensive. We want to be systematic. So how do we do that? There are some existing models, but most of them are based off of the pairwise relationships. So in this case, we have a target gene, we have a peak accessibility, and then this is one gene. You do a UMAP, and then the color represents how high or low the gene is expressed. This one tells you how open or closed this peak is. And then essentially these methods just do a very simple Spearman correlation test, to associate the peak to a gene. So in this case, you can link a cis regulatory element to the gene that it regulates. Okay. Another set of methods, instead of looking at a gene and look at the TF expression as well as the TF motif score. So the TF motif score, you can think of this as a average measure of how open or closed the TF binding motifs are along the genome. But again, this is based off of a pairwise relationship. You want to relate the TF expression to the TF accessibility element. But of course, like I told you, there are 3 different factors, okay? And then this is just one last piece of motivating or empirical evidence to suggest that we really should incorporate 3 different players in this model. So we have one gene, one peak, and one transcription factor. I don't know whether you can tell, but even from this UMAP, we see that they are all concentrated maybe in the natural killer cells as well as the CD8 memory T cells in this case, okay? So if you do a pairwise correlation between each 2 pairs out of these 3 different factors, you find that all of them are significant. As a matter of fact, the p-values are so significant. But of course, does it mean that these 3 different factors play together? So quite naturally, we should come up with sort of maybe a model that try to capture all 3 different players simultaneously. So as a very short recap, we have a gene, we have some potential candidate regions we want to scan for the enhancer regions, and then we have some TF that we know that are bound to the peak regions, okay? And then again, the existing methods are focused on the marginal and pairwise correlations between each pair of these 3 different factors. And there's this one method called PECA from Wingmall's lab at Stanford Statistics. So this method is primarily developed for the ENCODE data, where you have the bulk ATAC, actually the bulk DNase data as well as the bulk RNA-seq data. But importantly, we feel that this method makes a lot of linear and overly complicated assumptions, and it also is really hard to run a diagnosis. So in the end, what we really want to do is to propose a non-parametric, a model-free method for detect conditional association, okay? And then we really want to test how these conditional associations will return a cause that will complement with and corroborate with the existing methods that are purely based off of the marginal associations. Okay, so how do we do this? So the problems really just boil down to characterizing this function yg of xt and yj, okay? So the index g is for gene, the index j is for TF, the index t is for peak. Now each of this is actually a vector because you have the readout from all the single cells. Okay, and then we start with this one specific trail. So we don't consider cross TFs or cross peaks at this stage yet, because it's very similar to our GWAS studies, where you start with a SNP first, and then you think of ways to aggregate all the SNPs together, okay? So we focus on one trail for now, okay? And then there's a very simple linear regression framework, with or without the interaction terms. Your outcome variable is your gene expression. You want to test whether the gene expression can be explained by the TF expression as well as peak accessibility. But we know that the regulatory relationships are actually very complicated. It's certainly not linear. It can very well be nonlinear, or of a various form that we can just it's really hard to sort of quantify. So that's why we want to propose a non-parametric method. So how do we do that? We start by matching the cells by either their TF expression or peak accessibility. So in this case, we have a 3-dimensional sort of visualization. We have a gene expression as your outcome variable. We have 2 explanatory variables. Okay, so you match the cells by either their TF expression, and in this case, the green cells and the black cells will be matched together. And if you match the cells by their peak accessibility, now in this case for illustration, the black cell and the red cell will be matched together. Okay, and then the variable that is matched for, presumably will control for the variations in this variable. And then what we do next is to compute the differences in the other two variables that are not matched for. Okay, so you match by the TF extraction, you compute the differences in the delta G, YG, and you compute the difference in the delta Yj in this case. And quite naturally, just basically do a very simple experiment correlation test between the differences of the other two variables in this case. Okay, so you match by the TF extraction, you want to see whether there's a correlation while you already control for all the variances in the variable that is matched for. And then importantly, this matching really not only controls for the linear variation in the variable, but also the nonlinear variation as well. Okay, so this is what we call a level. Yeah.
So this may be a naive question, but exact matching may not always be possible.
Yeah, so you do need to relax that a little bit. Yeah.
And that will probably affect the performance, right? The level at which you match.
Well, yeah. Well, in this case, we don't really try to come up with something new. In this case, so we use some existing bipartite, matching. So you do a pairwise matching among all the cells that you have an observation for. Yeah. But it won't be sort of an exact readout, so we can't do that. Yeah. Okay, but this is level 1 testing. The level 2 testing is sort of an additional step of testing. You want to test whether this correlation is further dependent on the variable that is matched for. Okay, so it's almost like an interaction effect in your linear model in this case. But again, we feel that this is sort of without the linear assumptions. So without going into the algorithm details, I'm just going to sort of show you the package as well as the paper that has already been published. So again, in this case, we propose tripod. There are 3 different factors that we've been talking about. Okay, so back to the trail that I showed you before. So how does it look? So you do a level 2 testing, and then you find that the gamma from the level 2 testing is very significant. And then we feel that this is very strong evidence to tell us that these 3 different factors play together. The TF bind to the peak region, we regulate to this chain. Okay. Now, of course, we also look at some other data sets. So in this case, this is a mouse embryonic brain data set. And then the mouse, I think they are 15 days old. And then so this really is during the phase between neurogenesis and gliogenesis. And in this case, we resort to a very well characterized and very well defined set of trails. So using some biochemical and biological assays, in the 1980s, people have already found this U2 enhancer where the OLIG2 transferrin factor binds to this and then regulate to a very important gene in neurodevelopment that is called a SOX10. Okay, and then what we found is that through this genome-wide scanning, we are able to capture this relationship. And then most importantly, not only were we able to capture this relationship, represented as a blue arc in this case, we're also able to capture 2 additional arcs or 2 additional trail relationships. So one of them is easy, it's in the promoter regions. Okay, so we're not really interested in this. We also find another potential enhancer regions, and most importantly, these 2 enhancer regions are validated by the ChIP-seq. So basically, we know that the TF is actually bound at this region. Okay, so this is just one example. But so far, I've been mostly looking across the different cell types. How do we find the key cell types that this regulation sort of functions in? So we do a cell type specific regulation inference. And in this case, without going into the details, we propose a sampling-based approach to sort of identify the cell state or the cell types. And then this PBMC data set, we find, at least for this trail that I'm showing you, the CD8 and CD4 T cells, it seems to be the 2 cell types that this regulatory logic takes effect in. And then you can, of course, not only test for each individual cell types, you can test for multiple different cell types combined. So you first construct a hierarchy in this case, and then within each of the split as well as each of the leaf, you do a testing. And then the red dots here tell us that it is a significant enrichment. And then the footprint analysis from the ATAC part also confirmed that the T cells actually do play a major role in this case. Okay, so these are the discretized cell types. What if you have, I don't know, like a trajectory or maybe a developmental, cell states? And in this case, we're also able to, devise a method that are able to find a specific regions along the trajectories that are enriched for this regulatory logic. Okay, and then I'll just show you one very brief example. So in this case, we have a mouse hair follicle data set, and then there are 3 different sublineages, and then shown on the most right-hand side. And then for this trail, we find that it is most enriched for the topmost branch. And then again, the DNA's footprint analysis tells us that, this is confirms this finding. Well, of course, you can also find some additional enrichment in another lineage. In this case, I guess it's sort of very strong in the microlog, but maybe also in the hair shot as well. And you do find sort of a very convincing and a reaffirming evidence too. So how do we validate this? To our best knowledge, we don't think that there is an existing technique that can very easily validate this trivial relationship at high resolution and in a high throughput manner. Okay, so what we end up doing is that we try to adopt some existing databases or orthogonal sequencing studies that try to validate each pair among these three different factors. And hopefully, by doing so, we can complete the loops in this case, okay? So I'll just very briefly go over this. To validate that the peak actually is linked to the target gene, we first look at some super enhancer annotations. Now, in this case, you have some histone markers that are for the activation of the genes. You have the ATAC profiles for this specific cell type in the mouse hair follicle data set. You have a target gene. This is your favorite gene, for example. And then, the super enhancers for this gene has been reported previously in a Nature paper. And then we found that, the arcs that we found through tripod really just recapitulate what has been previously reported. So it's just one example, and then there are 4 additional examples as well. But of course, we understand that there's really not a consensus in the field in terms of how to best define super enhancers. So in addition to this, we'll also resort to some existing enhancer databases as well as cis eQTL databases. So in this case, basically, we carry out a hypergeometric test of enrichment. So we want to test whether there is a significant overlap between the calls from these existing databases as well as the calls that we return from our method. And then we benchmark against the 2 additional methods, and we find that tripod returns the most significant p-values of overlap in this case. Okay, so to validate the relationship between a transcription factor and a target gene, we utilize 2 databases. One is computational, the other one is experimental. And I'm just going to focus on this NOT-TF, which is sort of via a TF knockdown or knockout experiment. So you knock down or knock out the TF and you do a transcriptomic studies. And then very intuitively, you do a differential expression analysis. You can link the gene to the TF that you knock down or knock out, okay? And then in this case, we find that the conditional associations return higher precision as well as recall rates compared to the methods that are based on marginal associations. Okay, lastly, to test the relationship between the TF as well as the potential enhancer region, I've showed you some evidence of the DNA's footprinting, but we also look at some tissue and cell type specific ChIP-seq data as well. And then in this case, again, without going into the details, we find that we can significantly improve the accuracy in terms of predicting the binding of the TF in an enhancer region in this case, okay? So hopefully by doing so, we sort of complete the loop and then we show how what we call can corroborate and complement with what existing methods can do in this case. So this is a very busy slide. I'm not going to go into the details, but this is in collaboration with a pathologist at UNC. So basically, this is a single cell multiomic analysis of cells, actually cell lines as well as primary cells treated with HIV latency reversal reagents. And then we did some analyses, and then we have sort of a joint paper that is currently under review. And then we recently submitted a sort of a PO1 grant based off of the the results shown here. Okay. So that is the wrap of the first part of the talk. So I have to admit that most of this is really on the molecular level. We want to understand the mechanism of the regulation. Now, how is this related to health and disease? So during the second half of the talk, I think we're going to slightly shift the gear, and I'm going to talk about another framework that we try to relate genome-wide association studies with single cell data in this case. Okay, so yeah.
One question about the first part. So how would you apply your model if you only had transcriptional data? So for example, like we have a single cell atlas of the entire fly brain, but we don't have any of the cancer data. So are you confident enough in the model then that you you can apply it without any of the answer data?
I don't think so. You have to get the ATAC part. But I mean, you don't have to get the ATAC part from the same exact cells. In this case, I talk about the ATAC from the same exact cells, but there are techniques that can use to align the cells and to match the cells.
Even with the heterogeneity of the cells, then you think if you get a general ATAC for the fly brain?
Single cell ATAC, but it's just not single cell ATAC from the same exact cells. Yeah. So that's a good question.
So everything is focused on transcription activation, but can this also take into account repression?
That's an excellent question. I agree that most of what we do for now is focus on like a positive correlation coefficient. We're not really sure about the regression. We can call them, but it's really hard to validate them. And so we didn't really make a huge story out of it, to be honest. So, but I think that is definitely something that we need to look into for sure.
And I was thinking also about complexity of transcription factors. So that might change in different individual cells, but also can you accommodate that as well?
Yeah, so this is actually in my last slide, the future directions. So we need to do sort of a cross TFs and cross peaks. Very great questions. Yeah. So for now, it's just one TF, but definitely it's more than one TF in most cases. Yeah. Yeah.
So also network. So transcription factors can be expressed themselves, right, and feedback onto other transcription factors.
Yeah. Well, I also have the TS cascade, the temporal orders of the TFs in the last slide. So right on spot, like you need to look at the synergies between the different TFs for sure. Yeah. But are there any more questions? The second half is shorter, so it won't take as much time. All right. So more than a decade of genome-wide association studies really have identified these risk variants that are associated with complex traits. Okay, so each dot here is a locus or is a SNP, and then the height here tells us the strength of the association, the negative log p-values. So you identify all these variants that are associated with schizophrenia in this case. Now, emerging evidence really has suggested that these variants act in a tissue and cell type specific manner. Single cell RNA sequencing, as an example here, allow us to characterize cell type specific expression profiles across these different brain cell types. Okay, so the question that we want to go after is whether we can incorporate these two different information to prioritize trait relevant cell types, and therefore hopefully they can help us better understand disease etiology. Okay. This is sort of the end product. I want to show you this so that hopefully I won't run over time and then don't get to show you the very exciting part. You have the brain tissue, you have the other tissues. So this is a sanity check. For the schizophrenia, we know this is a positive control. These most likely will be a negative control because we know schizophrenia is something wrong with the brain, right? And then the method that we developed in light blue and the dark blue has significantly higher power in detecting the enrichment in the brain tissue, and it also controls for the type and error rate for the other tissues. Now, what's really interesting is that if you zoom into the brain cell types using single cell data sets, you are able to find the enrichment in these 4 different cell types, namely some excessive neurons, and then maybe GABAergic interneurons as well. And then we're able to reproduce these results using some independent cohorts as well. And then importantly, these also are in agreement with some recent reports in terms of cell type specific enrichment of schizophrenia. So the question is, how do we do this? Our goal here, without going into the existing methods, the pitfalls, the challenges, etc., I'll just show you this. We want to develop a biologically interpretable, statistically rigorous, and computationally efficient method to carry out this analysis. We know that the common variants and the rare variants should be modeled very much differently, and they perhaps also have very much different interpretations in biology as well. So we want to do this either separately or jointly for the common and the rare variants. We want to properly account for SNP and gene-gene correlations, as we feel that some of the existing methods are quite ad hoc. And then lastly, we want to go back to the gene level. So we want to help sort of prioritize genes in a cell type specific manner, okay? So our method is based off of a biologically interpretable model assumption and hypothesis from our point of view. And then the hypothesis is shown here. If a particular cell type influences a trait, then more of the GWAS polygenic signals will be enriched in genes with greater cell type specific gene expression. Now, this is very mouthful, so here is just one cartoon to give you sort of an idea, okay? On the y-axis, these are the GWAS gene-specific p-values. And then this is a specific cell type in the brain called the medium spiny neuron, MSN. And then on the x-axis, this is from the single-cell RNA-seq. And then you can rank the genes based off of their specific expression in MSN, okay? You can do some normalization, batch correction, etc. But you rank the gene for their specific expression in this cell type. Now, the hypothesis is that there is a positive correlation in this case between these two different parts. The more stronger the GWAS is, the more concentrated the expression will be in the MSN cell type. As a comparison, if we use a different GWAS trait, for example, the height, presumably having nothing to do with the brain cell types, then we see that there is, moderate or even no associations between these two different signals. Okay, so that is a hypothesis. How do we do this? We start with the GWAS summary statistics. We have the reference panel from PubMed or from 1000 Genomes Project. And then for the common and rare variants, we devise chi-square based test statistics for the common variants. And then we just use a very well-developed burden test statistics, which also is chi-square distributed under the null. And then nicely, we can combine these two together, as a chi-square with k+1 degree of freedom as well. Whether you combine them or not, or whether you model them separately, these will be used as your outcome variable y in this case. And then your features in this case will be your single cell type specific gene expression profiles. And you can incorporate some additional covariate, for example, the baseline expression as a correction. And then the epsilon term here, because all the genes are correlated, so you need to account for the gene correlation. So we use a generalized least square framework sort of to capture the correlation between the genes. Okay, so here is some additional evidence. So in addition to schizophrenia, we also look at bipolar disorders as well as SCGBIP. Again, we feel that the method retains higher statistical power, but also controls for false positives. Now, for the type 2 diabetes, here we use these two single-cell RNA sequencing studies from two different platforms. I believe the left-hand side, this is StopSeq. On the right-hand side, this is full transcript SmartSeq2. And then in this case, we're able to capture the enrichment in the beta cells as expected, because we know that the beta cells are involved in insulin secretion, etc. Okay, and then we do capture a gamma cell enrichment on the right-hand side as well. And then we feel that this also makes a lot of sense by talking to some domain experts. So what's really important in this case is that not only are we interested in capturing this enrichment in a specific cell type, but also we want to go back to the gene levels. We want to understand which set of genes actually drives this significant enrichment in the beta cells. So again, in this case, we basically use a very straightforward DF beta test statistics to test how much of an influence or how much of a leverage each individual observation, i.e. each individual gene has in leading to a significant enrichment of the beta cells for type 2 diabetes in this case. Okay, so we find all these different sets of genes. You do a gene pathway enrichment analysis, and then it's really reassuring that it seems that all of them are related to type 2 diabetes. And then importantly, you can overlay with what we call from this framework with what we can get using the each domain separately. Okay, and then I think the take-home message here, without going into the detail, is that they overlap, but they are definitely not exactly the same. So we feel that this framework allows us to offer sort of a prioritization strategy in a cell type specific manner. So how do we validate this? It's really hard. It's really hard to do a knockdown or knockout in a specific cell type in vivo in a living organism. Even if you can do that, the GWAS effect sizes are so small, you probably won't see anything in terms of their phenotypes after you do the experiment. So what we did in the end, as I'm going to show you one of the strategies, is to look at the existing publications by doing a PubMed search. So we basically just go through all the abstracts in PubMed, and then we search using a combination of the trait as well as a cell type, and we count the number of papers that actually have this in their abstract. And then the number of the publications that have these hits for both are shown on the y-axis. The x-axis are what we find as significant p-values, okay? So at least for the GTAC data, where we look at the bulk tissue enrichment, we're able to find a fairly decent Spearman correlation coefficient. Now, for the single cell part, it's not as impressive. Actually, it looks quite bad. We think that there is just not too many studies to date yet that try to find the cell types. But I think what's really reassuring is that we're able to find, at least for the GABAergic interneurons, we find the most hits from PubMed search. Okay, so this is one of the validation strategies that we carried out. So as good as this may all seem, there are some issues and pitfalls. To our best knowledge, almost all of the methods, including ours, focus on the SNPs that are in the gene coding regions or in the promoter regions, because we're talking about single cell RNA-seq. And then quite naturally, you need to unify the SNPs into a gene unit. So basically, you're just focusing on the coding regions and then ignore the non-coding regions. But we know that this is really what we're trying to understand in terms of their heritability as well as their function. Okay, so the solutions is single cell ATAC-seq or single cell RNA plus ATAC multiomic data sets. I have a slide, so this is our ongoing work with one of my PhD students, but I think I'm just going to skip this. But in short, we find very promising results, sort of using single cell ATAC-seq paired with GWAS summary statistics to do an unbiased scan along the genome, both coding and non-coding regions. So I've talked about these two different methods, but we've also worked on some other topics. So I studied my PhD on copy number detections, and then we also continued to work in this domain, extending it to the single cell setting. We have published several methods here. We also worked, fairly heavily on cancer genomics, so how to assess cancer heterogeneity, how to reconstruct the phylogeny of the cancer evolution. We worked on some methods that are specific for single cell RNA-seq. Some methods are specific for single cell ATAC-seq. So these are the methods that we developed as a lead author from me or my lab. But of course, in addition to this, we also collaborate with pathologists, biologists, clinicians. And here is just one example, working with Dr. Aziz Sankar. I think this is one of my most enjoyable and most fruitful collaborations since I started my lab at UNC. So together with him, we're interested in three different interrelated topics: DNA damage, repair, and circadian rhythm. And we now know very well that even from his Nobel lecture in 2015, that the repair of the DNA damage is actually controlled by circadian rhythmicity. And importantly, this control by the circadian rhythm is actually tissue and cell type specific. So going back to the theme of the presentation, we need to account for the heterogeneity when we do this type of assay. And then these are just some of the papers that we have jointly published over the years. Again, this is just a very pleasant experience. We do love to collaborate with people, and then we like to dive into a specific biological problems. And we really look forward to all the various different opportunities here. So with that, none of this would be possible without my wonderful students, as well as my mentors and collaborators, also the founding agent as well. So I think I can take questions from here. Thank you.
We're well on time. So we have time for questions.
Well, I have one more slide, so I guess maybe I can, talk about it. I still have 2 or 3 minutes to.
Yeah,
okay.
Sure.
So future directions. I'll just very briefly talk about this. What are we working on, and what we plan sort of on doing in the next several years? So for the second part of the talk, when you integrate the bulk tissue and the single cell data with GWAS, one of the things that we are interested in is to do a cell type specific eQTL or CAQTL. So this is expression QTL and content accessibility QTLs. But you do this in a cell type specific manner. Now we have aggregated data sets from various different donors for PBMCs, for fibroblasts, and for iPSCs from donors or donor lines. And then this is what we are currently pushing towards. Sort of we are trying to look at these massive single cell data sets, not just from one individual, but from 50 to 80 individuals. That is as as many as we can get at this stage. But I feel that the size of the data will really just grow exponentially from here on. And I think really what's really interesting in this case is how do you model this inter-individual variation? Where each individual will have an individual specific single cell variants in this case. Yeah.
So you brought up circadian rhythm and the changes that can happen. Would you also take that into account in the patient?
Yeah, there are some existing work sort of try to infer the phase for the circadian rhythmicity by single cell RNA sequencing that is published in Nature Communications. So definitely, I think that's one area that needs sort of more method and more attention for sure. Yeah.
Kind of a really different question. When you're developing methods, you can publish a paper and have your method spelled out and not actually have software that you use, all the way up to like making a really slick software that's easy for people. Kind of where do you fall with these methods that you're developing?
Well, we have open source and package for all of the methods that I talk about. People can ask questions via either Google user group, or nowadays I think everything is shifted to GitHub issues. So we do have we try our best to maintain it. Of course, we don't have a large group, so it's really hard. Yeah.
Are your methods in certain languages, or do you?
Mostly in R, but we do have some Python script as well as some shell script. Yeah.
So when you're looking at GWAS and single cell data, I imagine a problem is there's all these great GWAS in humans.
Yeah.
But then the single cell data in humans is very poor when it exists, right?
Yeah.
There are wonderful data sets in different species. So how do you are you comfortable? Can you use the mouse data with the human GWAS? And if so, can you use fly data with human GWAS? Where do you draw the line?
That's an excellent question. People have done that, looking at the homologous genes between human and mouse. So single cell data from the mouse and the GWAS from the human. But I think in that case, we just do only look at certain genes in that case, without considering any sort of evolution or anything like that. But yeah, I think certainly there's a lot of emphasis in this domain for sure. Yeah. Okay, so maybe I'll just quickly go over the next two without going into every single thing. So this is what Deb was asking for. So we're only looking at one trio at this time, but how do you look across different TFs or across different enhancer regions? Or how do you show the temporal orders of the TF? So this is one thing. And then as a matter of fact, there's also a very interesting observation that we find that the competent accessibility changes usually actually primes the changes in your gene expression. So what that means, we think in biology, is that usually epigenomic wise, you need to do some changes before it results in any variation in your gene expression in this case. And then we feel that the single cell multiomic data, because the cells are matched, it really allows us to sort of try to capture this lagging effect between these two different modalities. And then this eRNA profiling is what we are very excited about. At least as of now, we showed that the enhancer RNAs can actually be captured in terms of its expression and accessibility in a cell type specific manner by a very simple and standard 10x run. And we feel that this is an angle that we can sort of push towards. The enhancers, you can find cell type specific enhancers, for example, enhancer RNAs. But I think we do need some additional biological expertise in terms of trying to make a story out of this. Otherwise, it will most likely be a descriptive type of paper. Okay, and then the third part is cancer genomics and cancer heterogeneity. So one of my students literally just submitted a T32 yesterday, focused on this one. So we're trying to integrate bulk DNA-seq and single-cell RNA-seq, and we want to call the mutations in single-cell RNA-seq, and then reconstruct the phylogenies using both bulk and single-cell. But there are a lot of considerations, statistically and biologically, that we need to sort of be careful about when trying to do this joint modeling. And of course, by doing so, we feel that we're able to control for the DNA level confounding when we look at the RNA variation, or we can separate these different levels of variation. Then you can also do some deconvolution association testing. So I think that's all that I have. I hope it's not too overwhelming. Thank you so much. Yeah.
Any more questions for the speaker?
If not, let me thank Yuchao for a very nice talk and invite everybody for small lunch. Here next door is issuable room, and we have some fast sandwiches for lunch. So we can continue discussion with Yuchao there.
All right, thank you so much.